Hey everyone, this is Elad from Astrolabe Diagnostics, and today I'm going to talk about an interesting visualization challenge, namely how to present clusters to a researcher. Here is a common example from flow cytometry, mass cytometry, and single-cell RNA sequencing. You have a panel or a list of genes, and you use these to characterize different clusters. In this case, we're characterizing them based on rules from immunology. So we have B cells and different types of T cells and so on. However, the clusters might be anonymous. You know some of them are high for CD3, but you haven't called them T cells yet. In fact, you would like to have some sort of visualization that helps you identify them as T cells. One way to do it is using a heat map, as you see here, where every column is one of the markers, every row is one of the clusters or the cell subsets, and the intensity of the tile indicates the level or the expression value of that marker in that subset. And this makes sense if you really want all of this information. This is essentially a table where we replace the numbers with colors, and it allows you to relatively easily survey the different values. For example, if I want to find CD14 clusters, I can just go down this row and find them. Likewise for any other markers. And furthermore, there is a lot of white because most of these cells, most of these clusters do not express many of the markers in the panel. The challenge, though, is that we don't really have a relationship between these different clusters. In this case, in fact, they're sorted alphabetically, which seems to work nicely, but not exactly. For example, the plasmacytoid dendritic cells are down here, while the actual dendritic cells are over here. So the question is, how can we add a mapping or a visual element to the clusters that we have? One way to do it is to layer a dendrogram on top of the heat map, but dendrograms are a bit of a minefield. They're not always easy to navigate. One popular solution is using a minimum spanning tree, and this is used by both Spade and by Flowsum. So what you see here are the clusters as Spade sees them. So we take the high dimensional space and Spade breaks it down into a whole bunch of clusters. And here is how Flowsum sees the clusters. It is a grid that is part of the algorithm definition. Both of these methods use a minimum spanning tree to present the clusters. So the minimum spanning tree represents each cluster as one of these dots or bubbles, and they're connected using edges to each other. Now, a minimum spanning tree by its definition is a tree. And a tree is a graph, it's a term from computer science, where every two vertices or clusters are connected by exactly one path. And this means that if we have one cluster here and one cluster here, then there should be only one way to get between them. So for example, we cannot add this line to the tree because that would break the minimum spanning tree, the tree requirement. Another way to think about it is that trees have a starting point. They have a root. So for example, in this tree, this might be the root, and everything expands from that starting point. And trees have a lot of sense, make a lot of sense in the context of the immune system, because in many ways, development is a tree. Hematopoietic development is a tree. You start from a hematopoietic stem cell, and then the cells proliferate and differentiate into the different terminal subsets that we see in the immune system, such as the different T cell subsets. And importantly, there is no going back, so a T cell cannot revert and become a common lymphoid progenitor. And also, the pathways in the immune system are pretty much linear, which means that there is only one way to get to a given destination. There's a series of steps that the stem cell undergoes to become a CD8 positive T cell, and these are the steps that it will always take to get there. However, while this visualization makes sense biologically, the analogy starts to break when you look at actual experiments. And the reason is that your sample is not going to have all of these cells for at least two reasons. The first one is that there is no tissue in our body that actually has all of these cells plus the intermediates. For example, cells migrate from the bone marrow and then go develop in different immune tissues, and then they might migrate back to the bone marrow. So the bone marrow is going to have terminally differentiated T cells, but not the intermediates. And furthermore, most of the tissues in our body do not have stem cells. That is by design. And there are tissue-specific cells, such as microglia in the brain. So this is one way in which this breaks. A second way in which the analogy breaks, and this is relevant in the context of panel-based assays such as mass and flow cytometry, is that your panel might not have the markers that you need to identify the intermediates properly. And your panel might not have the markers you need to identify a given subset at all. So it's possible that your panel doesn't have any markers for T cells, and yet your sample might have T cells, and that might interfere with the logic that goes into the minimum spanning tree. And in fact, researchers who use these tools on a regular basis are familiar in the ways in which this logic can break. And this is the same figure we've seen before from the Flowstone paper. And you can see here that the CD4 T cells are in light blue, and then they branch into NKT cells, which in turn branch into NK cells. Now, biologically, this does not make any sense. I will clarify that from a visualization perspective, this is fine. We get an idea of what's going on. We can color code this by different markers, and we will get a good representation of the different cell subsets. If our goal is to label clusters, then this could work. However, it could lead to some confusion. And furthermore, it is common to see these weird situations where a given cell subset might break, and one half of it will go into one branch and the other half would go into another branch, and there might not be biological meaning behind it. It's just fluctuation or noise with the data. So here is another suggestion. This is an algorithm called multidimensional scaling. It's a method that's been around for several decades now, and in many ways, multidimensional scaling is the older sister to principal component analysis, or PCA. In multidimensional scaling, we supply the method with the distances between all of our points or all of our clusters. And this is a very common example from multidimensional scaling, where the table that we inputted to the method are the distances between cities in the United States. And you will notice that the output, the map that MDS gives us, is a fairly accurate representation of the country, of the United States. There's a few mistakes. For example, Buffalo is way too south. However, the general structure is pretty similar. Multidimensional scaling, it belongs to the family of algorithms known as dimensionality reduction algorithms. Another common example that is used in flow and mass cytometry is t-SNE, and UMAP is another one. Conceptually, the big difference between MDS and t-SNE is that MDS is a linear method, while t-SNE and UMAP are nonlinear. However, when talking about a handful of clusters, these differences empirically often do not matter. When going into single cells, the distinction between linear and nonlinear methods is quite critical. However, when just visualizing 20 or 30 or 100 clusters, the linearity restriction on MDS is usually not a problem, and you can end up with very nice maps. And here is an example from the immune system, and I'm using the Astrolabe platform to demonstrate this. We're seeing an MDS map for one of the experiments that we have, and you can see the different bubbles correspond to different cell subsets, and their position on the map correlate with the high dimensional distances. So all of the T cells are next to each other, and over here to the right, and then the B cells are far away down here, and the myeloids are up there. So in effect, we're maintaining a coherent structure. There is no limitation of a tree, and there is no edges connection in the bubble because there is no graph underlying this. And yet, the map that we get makes sense and allows us to interact with the data. And like many of the other methods, we can color code this. For example, here's all the CD3 positive bubbles, which corresponds to T cells. And here are the CD20 positive bubbles, and the plasmablasts are lower than the rest, and so on and so forth. I hope this demonstrates the different tools that you might have when you want to visualize your clusters. Again, you can use a heat map, you can use a minimum spanning tree, and I would like to propose the MDS map or multidimensional scaling as another candidate. As always, if you have any questions about data analysis, if you need help analyzing your experiment, please reach out to us at Astrolab Diagnostics. And please subscribe to our YouTube channel and to LinkedIn to get more of these videos and more of these ideas on how to analyze your data. Thank you and have a great day.